Okay, let's go ahead and get started now. I've got everybody in here. We're going to take a look at cell division and mitosis today in lab. We're also going to take a look at what's known as diffusion and osmosis, movement across membranes. So both of these were found in the eLearn content. Anybody have any questions about anything with lab or the lab exams coming up or stuff like that? Now we do have, next week is when I'm going to open up the lab exam. We will have regular lab that day. We'll start the bone unit, the skeletal system. We'll start looking at that, and we will. But I'll open up the unit test 1 next week, and I'll leave it open for 10 days. And you'll have the test will be 50 questions. You'll have an hour to take it, and it'll all be randomized. And but in the meantime, we're going to go on. The sooner we get stuff done, the sooner we get the lab done. So let's see what we have. You're correct. There are no lab assignments or lab reports in here. Even if we were on the ground, there would be no lab assignments or lab reports. You're just responsible for the content we cover, and that's all. So, and nobody does lab assignments or lab reports, any of the anatomy labs on any of the campuses. It just there's so much to try and master that it really is an extra amount of work. The exam will be multiple choice. It'll be 50 questions, multiple choice. There will be pictures embedded in the exam, and hopefully they work fairly well. And a lot of it will be identification, the various slides. So if you can go through and be able to identify all the epithelial cells and all the connective tissue and the muscle cells and the nerve cells, you can identify that in the parts of the microscope. That's a big hunk of the test right there. So, but it will all be multiple choice. Now, I will say that the multiple choice questions will have lots of answers. It won't just be 5 answers. It'll probably likely be closer to 25 answers to choose from. Since, doing a lab exam in multiple choice is somewhat challenging. So, no, you will not be able to use your notes for the test. You won't have time to go through the notes. I intend to go over that very carefully because I'm not happy of the. I have better pictures of the stages of mitosis in the PowerPoints here. So. So yeah, and to answer your question about assignments again, nobody has time to do in-depth homework assignments. It's enough to try and keep up with all the classes. I have 4 lectures and 4 labs to cover. And the only advantage that being online gives me is that it saves classroom time. That's the only advantage I've had. This is the first year that I've ever actually gotten done on time. Because usually there's so much to cover in class that none of us can get to it. So anyway, that's enough of that. Okay, so let's go ahead and get into this. And I will try very hard to make it as interesting as I can. And so there is actually some relevance to this. Let's take a look at, here we go. The cell cycle and mitosis. So you should all see this screen here that says the cell cycle and mitosis. I'm gonna start this now. Okay, when we look at the cell, now this is something we're gonna cover more in much greater detail in lecture, but our cells are the smallest unit of life. Nothing smaller than a cell is alive. Every living cell comes from other living cells. There are no bottles of Insta-Life in the lab where we can create artificial cells. And we can make an artificial cell out of a plastic bag, and it will allow water to go through. It has little tiny pores in the bag, but it's not alive. Only living cells only come from other living cells. Now, in our bodies, we have a wide variety of cells. We have billions of cells, and they're very specialized. You saw that last week looking at the epithelial, the muscle, the nerve, and the connective tissue. So we know that there are lots of types of slides. Some of those cells are very, very long. Neurons can be 3 or 4 feet long. There is a nerve in our body called the sciatic nerve. It starts at our hip and goes up down to our big toe, runs behind the knee, and it's about the thickness of your thumb, and it is about 3 or 4 feet long. And those are made up of, the nerve is made up of thousands of individual nerve cells. So we can have cells that run a great length. We can have very short cells. We can have little tiny columnar cells lining our small intestine. We can have muscle cells and nerve cells of different shapes and sizes. Most cells will reproduce. Some don't. Most cells, nerve cells and muscle cells don't reproduce. The nerve cells and muscle cells we have when we were born are what we have now. They can only wear out. They can't replace themselves. Most cells are going to have all the same structures. They're all going to have a nucleus. They're all going to have DNA. They're all going to have a cell membrane. The only cells that don't have a nucleus are the red blood cells, because red blood cells don't reproduce at all. They'll get made and last for 4 months, and then they'll be broken down and recycled. This is a cell. This is a cell that sort of has everything attached to it. Not every cell looks like this, but they look pretty close to it. If you're a cell, you're going to have 3 parts. You're going to have a nucleus. You're going to have a cell membrane, and you're going to have the cytoplasm. Now, red cells, again, are the exception. They have no nucleus. They don't have any of these what we call organelles inside here. They're just packed full of hemoglobin. They're a big squishy sack of hemoglobin designed to carry oxygen, and that's all they do. But all the other cells in our bodies have all these structures here called organelles. The nucleus is an organelle. The green structure here called the Golgi Apparatus is an organelle. These little things that look like sausages are mitochondria. They're organelles. We can have, some of our cells have villi in here, little folds that stick out on the edge of the cell membrane to increase the surface area. Those simple columnar cells that line the small intestine where we absorb nutrients have villi and microvilli that stick out to increase their absorptive area. In fact, the small intestine is typically about 20 feet long and about big around as your finger. And yet, if you were to stretch it out flat, it would cover, if you stretched out to one cell layer thickness of the small intestine, it would cover a tennis court. So it's a huge amount of surface area. So the nucleus is the control center of our cells. It has where the DNA is located. Every cell except the red blood cells has a nucleus and has DNA. The DNA in every cell is identical. It's just what parts, for different cells, different parts in the DNA is turned on and other parts are turned off. The DNA is scattered around when it's unwound into something called chromatin. When the chromatin condenses, it forms chromosomes. We're familiar with the term chromosome. Genes are nothing, you've heard the term genes. Genes are the physical location of specialized areas of DNA that control traits. So the gene for eye color is a segment of DNA that produces the pigmentation in our irises. That's a gene. And every, and our DNA will have that information, or hair color, or blood type, or body stature, or whether you can curl your tongue or not, believe it or not, or whether your earlobes attach or dangle. Those are all traits. And the traits are controlled by our genes, and our genes are nothing more than the physical location on the DNA of the information for that particular trait. So it sort of takes the luster off the term gene when you realize it's just, it's here, that's all. Now cells are a typical living cell has what runs through a well-defined cycle. And it's cleverly called the cell cycle. There are two parts to the cell cycle. One phase is called mitosis. And mitosis is known as nuclear replication. Nuclear replication. We make a complete copy of all the chromosomes and all the DNA in the nucleus, and we produce two identical sets, so we can make two daughter cells. Now, mitosis and cell division are two separate things, because if mitosis occurs without cell division, you get one big cell with two nuclei. You got to have cell division too. Cell division is separate from mitosis. And while that doesn't sound, while it sounds like we are splitting hairs on this, for testing purposes, at least in the Walter State Natural Science Division, if you see a question on a test that says mitosis is, mitosis is, and it says cell division, that's wrong. Always mark nuclear replication. So anyway, the cell cycle consists of two phases. Mitosis, where we replicate the DNA, nuclear material, and interphase. And interphase is everything else. Interphase is what a cell does when it's busy being a cell. When the cell is busy being a cell, doing cell things, absorbing nutrients, growing more bone, sending signals releasing hormones, things like that. The whole time a cell is busy being a cell is interphase. Now we will duplicate our DNA in interphase, but that's as we get near the mitosis stage. But we spend most of our time as a cell in interphase. Now I know at some point mitosis is often taught as the stages of mitosis are interphase, prophase, metaphase, anaphase, and telophase. Interphase is not part of mitosis. Interphase is the 99.99% of the time the cell is busy doing cell stuff. Mitosis only takes a few seconds. Interphase is when the cell is doing its job, however long that takes. So, okay, so here's what interphase looks like. Here's a cell in interphase. Cell doesn't look like it's doing anything. It isn't now because it's been put on a slide. But when it was alive, it was busy doing cell stuff. The DNA inside the nucleus was controlling the activities of that particular cell. And whether it was a columnar cell, which is sort of what it looks like, but I'm not certain, whatever that cell was doing, the DNA was controlling that. Now, as we move through the cell cycle, the overarching cycle of the cell is called interphase. And there are various stages of the cell cycle. We'll get into all those stages in lecture. But as we move through those phases, those stages of interphase, different things happen. And part of what we do is we do replicate the DNA in anticipation of going into mitosis. Because cells, cells will wear out. And when a cell starts to wear out, it will just decide it's time for mitosis to occur. Now, if a cell lives for 10 days, it will spend the last 30 seconds of its life replicating itself, making, it will separate the DNA, it will copy the DNA and then make 2 daughter cells. And then, you've got 2 new cells going. But again, the amount of time spent in interphase is probably 99.99% of the cell. Mitosis lasts seconds. Now, this is an example. Here we see cells dividing, undergoing mitosis and cell division. Now, this is known as cytokinesis, when the cell physically separates. At the stages of mitosis, this one is particularly good because it's very quick. From start to finish, mitosis is very rapid. It doesn't last for hours or days. It's over in seconds. And if we have cytokinesis, we get 2 distinct daughter cells. We only get daughter cells if we have cytokinesis. Otherwise, we get a great big cell with 2 big nuclei, and that's not going to work. So the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. These are the 4 stages of mitosis. Interphase is not part of mitosis. Interphase is when a cell is just being busy being a cell. Prophase. A lot of things happen in prophase. Now, we've taken our DNA, and somewhere during interphase, we've copied our DNA because we have to have a second set of DNA to make 2 daughter cells. So at some point in interphase, prior to entering mitosis, we have copied our DNA. Now in prophase, a lot of things happen at once. All the DNA that's been copied and the DNA that was, both sets of DNA start to condense. All that chromatin comes together and condenses and forms the chromosomes. And the chromosomes are called chromatids. You have 2 chromatids held by a structure known as a centromere. We form what are known as the spindle apparatus. There's structures in the cell called centrioles that form, we call them spindle fibers. You probably remember that from some point in an earlier science class. Start forming these fibers that stretch across from one side of the cell to the other. We see the loss of the membrane around the nucleus, called the nuclear envelope. So if you look at this picture here, what you're seeing, all these black squiggly lines, those are the condensed chromosomes, but they're not organized. The nucleus is missing. See, the cell next to it has it, there's its nucleus, and the cell right next to the red circle, and the one above it has its nucleus. In fact, many of the cells here have a nucleus present, but the one in the red circle doesn't. The nuclear membrane is gone. The chromatin has condensed into chromosomes, and they look very disorganized. but this is a planned event. Once the nuclear membrane is gone and spindle fibers start to form, the chromosomes that are visible now start being moved towards the middle of the cell, to the midline of the cell. This is the second stage of mitosis. This is called metaphase. And in metaphase, the chromosomes are pushed and pulled to the middle of the cell by the spindle fibers. Spindle fibers attach to the centromere. And they're pushed to the middle of the cell. And the question, of course, is why they go to the middle of the cell? Why do they do this stage? And this is to make sure that we have even separation. So that, you know, when the chromosomes are pulled apart and pushed apart, you're going to have, you know, 23 pairs of chromosomes going to the left side and 23 pairs going to the right side, because you've copied the DNA. And you want to end up with daughter cells, with each daughter cell having a complete copy of the original DNA. So that's why they line up like this. They are pushed and shoved and dragged by the spindle fibers. Some of the spindle fibers attach to the chromosomes and pull them. Some attach and push them. Some are just there to push the cytoplasm apart. So, and you can see, if you look carefully, here we see on the red one, we see the chromosomes present there, but here we see in the red circle, we can see some of the fibers. And that line, that dark line there, are the chromosomes, and we see some of the spindle fibers moving apart. Now this next stage, once the chromosomes line up, then we start to move them. We start to move them to opposite sides of the cell. Now we're still one big cell. You know, in metaphase, we're still one big cell, but now we've got the chromosomes. They're lined up, they're evenly distributed, and now they're ready to move. Because if they're not evenly distributed, nothing's going to happen. So when they're lined up and evenly distributed, they start to move, and that takes us into the shortest stage of mitosis, and that's anaphase. The paired chromatids split. You can see the spindle fibers now, right? You can see them in the middle of the red circle. Those are the lines. They are pulling and pushing the individual chromatids. The chromatids split apart. One chromatid going to one side, one chromatid going to the other side. You end up with a complete set of chromosomes from both sides. Now, anaphase is the shortest of the phases. It happens so fast, you can barely see it, because we go from metaphase to the last stage of mitosis, telophase, very quickly. So the chromosomes move apart rapidly. And once they reach the opposite ends of the cell, then they stop moving, we're going to move into the last stage called telophase. Now, the actual physical separation of the cell starts in anaphase. We have the process known as cytokinesis. You can see it up here. It's the third bullet down. Cytokinesis, that is the physical separation. We have a ring of protein, what we call contractile protein. It's the same protein that we find in our skeletal muscles that allow our muscles to contract. That band of protein will start squeezing from the outside and physically separate the parent cell into two daughter cells once the chromosomes have been fully moved to opposite sides. And this stage is telophase. The last stage is telophase. We start forming a new nuclear membrane. The spindle fibers are going to disappear. The chromosomes uncondense and become chromatin again. The nuclear membrane is fully formed again. the nucleolus, a structure within the nucleus, reforms. And we have the end of cytokinesis. You can see now, this one's not completely separated here because there's still a line that hasn't formed here. But now here, the one in the red oval, you can see that cytokinesis is just about completely finished. You know, we still have some spindle fibers to get rid of, to reabsorb, but the cells are just about finished. And then we see in this last slide, cytokinesis is complete. We have two distinct daughter cells. The plasma membrane is pinched in half by the protein, and we have two distinct daughter cells with two sets of chromosomes, which are identical to the parent cell, and they're going to live as long as, you know, are they 10 days? Are they 2 weeks? Are they 3 days? Are they 20 years? It just depends. And when they reproduce, they will follow this same pattern in the cell cycle. And we'll discuss the cell cycle in much greater detail in lecture. There's a lot more to it than this, but this is basically the stages of mitosis and cytokinesis. Now, as far as testing goes, if the study guide doesn't have great pictures, you can take a look at the PowerPoints. I think the PowerPoints have pretty good representation here. I will tell you, for testing purposes, I have tried to use the very best pictures of mitosis and cell division that I could. So you shouldn't have any kind of, you know, fuzzy looking pictures in here. So should have better quality pictures. Okay, so that is cytokinesis and mitosis. And of course, we'll have a lot more to look at when we get into lecture on that. But this is the basis of it. This is, you know, so let me get out of this. Anybody have any questions about this? Did I answer your question about the stages of mitosis?
Yes, you did. Thank you.
Good, good. So now, if you're comfortable with that, you know, I felt that the slides here would address that fairly well. So, okay, so let's go into, let me go into share screen again, and now go into the other aspect I want to cover. Membrane transport, here we go. The other thing we want to talk about a little bit is membrane transport and permeability. I want to introduce this now because you'll hear about it again in chemistry, in lecture, and in the cell chapter too. Cells have the ability to move stuff in and out of them all the time. Membrane transport. Cells are surrounded by a membrane. All of our cells have a membrane. And that membrane is what we say selectively permeable. It is very picky about what it lets through. It does not let through anything that's water soluble, at least not easily. Since the membrane is made up of a lipid barrier, things that are fat soluble, oil soluble, slide right through. Hormones that are made from cholesterol, the steroid hormones, the sex hormones, for example, all slide right through. But anything that's water-based has a real hard time getting through. So we have to understand how cells work to get nutrients inside and to get waste products out. Now, when we say cells are covered with interstitial fluid from the blood, what that means is that our blood vessels are very, very leaky. Our blood vessels have tiny holes in them lots of them, and they leak fluid all the time. Plasma leaks out from our blood vessels all the time. That's what they do. And they're supposed to do that. This interstitial, we call this fluid in the blood the plasma. That's the salt water that the red cells and the white cells swim in, if they swim, as well as salts and nutrients and hormones and stuff like that. So the plasma will leak out. When it leaks out, it becomes interstitial fluid. That means it's the fluid around the outside of the cells. It doesn't get into the cells, it's outside the cells. Now that fluid contains things the cells need. It contains nutrients, it contains hormones, it contains sodium and potassium and fats. These are things the cells want, but they're only going to take what they need, not just take it all in. This fluid is then collected by our lymphatic system. Our lymphatic system is what we call a parallel circulatory system. So all this fluid is passively collected by our lymphatic system. We run it through our lymph nodes where our white blood cells hang out. Our white blood cells are in there looking for something to kill. They are monitoring our lymph, looking for any kind of pathogen. Once the lymphatic fluid is determined to be pathogen-free, it is then returned to our plasma. So plasma becomes interstitial fluid, which becomes lymph, which becomes plasma. It cycles around over and over again. Cells love to not spend any energy to get stuff inside their inside. They love passive movement. And they don't like to have to pump things in. Anything that can come into the cell for free is a great deal as far as the cell goes. The only time that they will spend energy is when they want to get something in that has a hard time getting across the membrane. So, transport and permeability. So if we look at this next slide here, there are three types of movement across a membrane. We have what's known as, and this is a cell membrane here. It's made of little fat particles. So we say it is hydrophobic. It hates water. Now, we call it a lipid bilayer because it really is a two-layer structure made up of fats. And some things will slide through the membrane easily. We call this diffusion. Diffusion is movement from a high concentration to a low concentration. That's what's happening here. We see all these little purple spots. Well, they're gradually moving. I think they're purple. They're moving across the membrane to the inside of the cell. It's passive. We go from a high concentration to a low concentration. It's like it's going downhill. Here we see, this is known as facilitated diffusion, where we have a special channel in the membrane. This special channel will let certain things come through the membrane. The only, whatever's out here, these little orange triangles, can't get across the membrane on their own. The cell won't let them in. But there happens to be a special channel there just for those little orange triangles. All orange triangles come over here. Here's your opening. And we go through that special channel, and only orange triangles can go through there. Purple circles can't go through. Green squares can't go through. Only orange triangles. That's a special channel there. In many cases, we call it a carrier protein. It's usually a tube through the membrane made out of protein. So, and this is sort of what happens with insulin. Insulin that we release when our blood sugar goes up, we want to get that sugar into the cells. Insulin stimulates cells to make more of these we call them carrier proteins. So we get what's known as facilitated diffusion. We can diffuse from a high concentration to a low concentration. Like if this is sugar out here in the interstitial fluid, if this is sugar, we can get that sugar into the cell from a high concentration to a low concentration, thanks to that particular channel that opened up for us. Here's the third type of membrane transport. This is known as active transport. Active transport is essentially going uphill. The cell has to pump something across its membrane against its concentration. You'll notice that there's a whole bunch of green squares down here and only a few here. Diffusion would say the green squares should go out. The green squares shouldn't come in because the high concentration of green squares is on the inside. Instead, the cell says, I need those green squares, and it will actually pump across the membrane against its concentration gradient, meaning it's pumping from, instead of going from high to low, it's going from low to high. It's going uphill. And that takes energy. And this little yellow thing here says ATP. ATP is the molecule that we cells use, that our bodies use to carry energy around to all the parts of our body to get things done. So ATP is the fuel that we use to power our muscles and nerves and everything else. And so this is actively transporting the green squares against the concentration gradient to get inside the cell. And later on, I'll tell you what the green squares and triangles are, stuff like that. So, okay. Two types of transport across membranes, active and passive. Cells love passive. They prefer to spend no energy at all to get stuff across the membrane. So, diffusion and facilitated diffusion are great. From high concentration to low concentration. Slide right into the cell. Even if you have to have some sort of special channel to get in, that's still a free ride. Active transport, it must be something that's really important to the cell because it's going to have to pump it against its concentration to get it in. And it will spend the resources, it will spend the ATP to get stuff inside the cell. Now, what are we talking about getting into and out of the cell? Well, to understand that, we have to understand what a solution is. And you'll hear about that in lecture, probably on Thursday. You'll hear all about solutions. A solution is a mixture, usually is a mixture of a solvent and a solute. Usually water is the solvent, and who knows what the solute is. If you're going to make salt water, you're going to make a glass of salt water. I don't know what you're going to make, but you just want to make a glass of salt water. Okay, so you take a glass of water and dump in a couple of tablespoons of salt. The water is the solvent. It is the dissolving medium. The solute is the substance that went into solution. It disappeared. You put some tablespoons of salt into water, and the salt disappears. It's still there, but it's, as we say, in solution. And now you have a salt water solution. It is clear, it is transparent, it is what we say homogeneous. It does not, you can't see the water and the salts. The water and salt don't appear separately. You see a transparent, you only see the water. You see what's known as a transparent solution. a solvent and a solute. Whether you're making sugar, you're adding sugar to tea, you're making sugar water, maybe you have a hummingbird feeder, you've got sugar water for that, maybe you are making salt water for whatever, and you have a solution. It's transparent. All solutions are transparent. Whether it is a Coke, which is transparent, it's a solution, even though it has the coloring in it. It's still, you can still see through it. Or coffee or tea, you can still see through that. It is still a solution. Solvents and solutes. Now, most of the water and fluid in our bodies are solutions. The plasma is by itself is a solution. It's a mixture of water and salts and proteins and fats and hormones and vitamins, all this other stuff in solution. The fluid inside the cell and outside the cell are solutions. We have what's known as if we had interstitial fluid earlier, we have the salt water that surrounds the cells. Where'd that come from? It leaked out of the blood. It was plasma that leaked out. It's a solution. The intracellular fluid, the fluid inside the cells, are also a solution. There's essentially no difference between the cytoplasm and the interstitial fluid. It's the same, essentially, fluid. It's a solution. Now, our membranes, our cell membranes, are very, very picky about what comes across. We say they are selectively permeable. I mentioned this earlier. We have to have specialized mechanisms to get some things across. Other things slide across very easily. If you think about, sort of digress off this, think about Italian dressing, salad dressing. What do you have to do with it before you use it? You have to shake it up, because if you don't shake it up, you've got the oil layer and the vinegar layer. And the oil layer and the vinegar layer, because the vinegar layer is mostly water, they don't mix. And this is essentially what's going on with the cell. If you had a cell, you would get the oil layer to slide right through the cell. The vinegar layer wouldn't. The cell is selectively permeable. Some things it lets in, it excludes other things. So this selective permeability, and that's going to affect diffusion and osmosis. So diffusion is the movement from a high concentration to a low concentration. Here we see a beaker, and I've dropped a pellet of dye in the beaker. I actually do this in class, where we put a dye, a couple crystals of methylene blue into a beaker, and it dissolves in water and it's highly concentrated when it first goes in, and then gradually spreads. The high concentration, of course, is here, and now we've reached what's known as equilibrium. When the coloring agent is distributed equally throughout the water in the beaker, we've reached equilibrium. But until then, we move from a high concentration to a low concentration. That's what happens in our cells with diffusion. We move from a high concentration to a low concentration. We are trying to establish equilibrium. We're trying to move something into a cell to reach a level where we're equally distributed outside and inside the cell. Unfortunately, for us, we can never reach equilibrium. We don't want to reach equilibrium, so that stuff tends to come in, or stuff leaves the cell and goes out, because diffusion can work both ways. Now, a little more specialized discussion here. Diffusion is the movement from always the movement from a high concentration to a low concentration. It is random. It is passing. Molecules move randomly. They bounce around. And so they will gradually move from their high level to their low level until they reach equilibrium. You know, in class, I've been known to open up a bottle of some real cheap, you know, air freshener or something in one corner of the lab and open it up and wave it around a little bit, maybe spray it around in the corner, and then let it spread throughout the room. And eventually it diffuses to where everybody smells it. You know, I try to pick something that isn't too offensive, you know. But we diffuse from a high concentration to a low concentration until equilibrium is reached. As I said, fortunately for our cells, we never reach equilibrium because we always want diffusion to work for us. Now, diffusion works across a membrane if the substance is lipid soluble. Will it dissolve in fat? Diffusion works that way. Diffusion works if there are already channels available in the cell membrane. Water moves into cells because there are special channels just for water. Or is there a specialized carrier molecule? Glucose requires a special carrier to get in. Water can come through, the little channels called aquaporins, because water certainly isn't fat soluble. And water is normally kept out of the cell, but there are channels for water to come through. So water can come into the cell through these channels. Glucose gets into the cell, but it has to have help. It has to have some sort of carrier molecule to get in. So it is facilitated. Now, simple diffusion works all the time. You know, hormones that are made of cholesterol slide through. Anything that's soluble in fat, anything that cholesterol slides across the cell membrane. You know, so all this will come into the cell very easily because anything that dissolves in fat will go through the cell membrane. So that is, you know, if it's also the other thing that will get in is very small particles. Oxygen diffuses into the cell with no trouble whatsoever because oxygen molecules are very small and they will come through the cell membrane and diffuse. And we always have a higher concentration of oxygen outside the cell than inside. We never want to have equilibrium because we always want more oxygen to come in. Carbon dioxide will always diffuse out of a cell because we always have greater CO2 outside than inside. We don't want to have equilibrium there either. We want to have CO2 always leaving the cell. You know, small, you know, oxygen's not fat soluble, but oxygen goes across the membrane very easily because it's tiny. Things will go across if they're tiny, if they're fat soluble, if there are special channels, or if there are carrier molecules. So simple diffusion is totally unassisted. Anything small enough to go through the membrane or small enough to go through the specialized pores in here. Simple diffusion. It's either fat soluble or it's very tiny. So we have that advantage there. Now there is a specialized type of diffusion that involves the movement of water across the membrane. And it's called osmosis. You may have heard of this in the past. Osmosis is the easiest definition for osmosis is the movement of water across a membrane and it has to be across the membrane. Movement of water across the membrane from high water to low water. Water will move from the high water concentration to the low water concentration. High water to low water. For example, here we see an example. We have a beaker with a membrane in the middle. On one side of the beaker, on one side of the membrane, we have a solution that is mostly water with a little bit of sugar. So it says low sugar concentration, high water concentration. So the low sugar concentration is, let's say it is 10% sugar on the left-hand side, which means it's 90% water. That would be a low sugar concentration. 10% sugar, 90% water. The other side of the membrane is 90% sugar and 10% water. So that's sugar solution on the other side. It's like a slug, but we'll just leave it like that. High sugar concentration on the other side of the membrane. On the right-hand side, this is 90% sugar, 10% water. Over here on the left side, it's 90% water and 10% sugar. 90% water is significantly greater than 10% water. 90%, if the water concentration is high, then the solute concentration is low. The water concentration is high, the solute concentration is always low. So you always go from the high water to the low water. In this case, on the left-hand side, the water concentration is 90%. On the right-hand side, the water concentration is 10%. So water will move across this membrane from the left-hand side to the right-hand side. This is osmosis. Water moves from high water to low water. Always remember, write that down. Always remember that water moves from high water to low water. High water equals low solute. Low water equals high solute. Water always will move across the membrane from the high water side to the low water side. In lab, we do it when we're on the ground, we do a demonstration. We take an artificial cell made out of a plastic membrane. It's called dialysis tubing, and the dialysis tubing has little tiny holes in it. It's used in dialysis treatment of kidney patients. If you make an artificial cell out of that dialysis bag and fill it with 10% glucose, no, we're going to make it 40% glucose, 60% water, 40% glucose, and put that artificial cell into a beaker of distilled water, 100% water. You weigh the cell before you put it in, and then stick it in the beaker for 2 hours, and then take it out and weigh it. You will find that it gained weight because water will go through the membrane. Because if you're in pure water, it's 100% water. The contents of the bag of the artificial cell was only 60% water. The higher side, the water side that was highest, was outside the cell. Water went into the cell. It gained weight, gained water. And that's osmosis. High water to low water. It is a type of diffusion. Okay. This is an example of diffusion through a different type of medium. Here we take an artificial, well, we take a petri dish and we put a crystal of methylene blue and we put a crystal of potassium permanganate on here. And we wait and we see what happens. And we discover that both of these substances will diffuse across the gel. Now, one thing we discover is that movement across the membrane is assisted by the size of the particle. Small particles, small molecules go across the membrane faster. or they go through the medium faster. In this case, the medium is a gel, which makes it even more challenging. But potassium permanganate molecules are smaller than methylene blue particles. And so potassium permanganate spreads out faster, than methylene blue does. So particle size helps to determine the rate of diffusion. The smaller the particle, the faster something will diffuse. Temperature also plays an effect here. Things diffuse faster at higher temperatures. The faster the particles are moving, the faster they're going to diffuse. If I had placed this on a hot plate that was just warm enough to keep the agar warm without melting the agar, because the agar is sort of like Jello, I would see that the permanganate would go across the agar much faster than it was doing at room temperature. So particle size and temperature affect diffusion, as does concentration. The more particles you have, the faster it's going to go. It's going to diffuse. So don't do that, don't do that. And this is what I was talking about. These dialysis bags. Here's an example of the osmosis I was telling you about. Here you have these bags of dialysis tubing filled with a solution. One of them has 10% sodium chloride in the beaker, and the other beaker is filled with distilled water. And the dialysis bag, in the case of beaker 1, the dialysis bag would have 100% water, and in beaker number 2, the dialysis bag would be 10% salt. And so what would happen is, since the beaker is 10% salt and the bag is 100% water, the bag would lose weight as water came out. In number 2, the beaker is 100% water. The dialysis bag is 10% salt, 90% water. And so water would go into the bag. And of course, we demonstrate that by weighing everything. And we find that if we put 100% distilled water in a dialysis bag in a beaker full of 10% sodium chloride, osmosis works on this. And so the water in the dialysis bag will leave the bag and go out to the beaker, because inside the bag is 100% water. Inside the beaker, it's only 90% water. Water goes from high water to low water. That bag will lose weight. Here we have just the opposite. We have the saltwater solution in the bag and 100% water in the beaker. The higher water concentration is outside the beaker, and it will move across the membrane of the dialysis bag, and that bag will gain weight. And so this slide shouldn't be here. Okay, so this is diffusion and osmosis. I hope I didn't confuse you too much with it. Basically, you need to be able to remember diffusion, high concentration and low concentration. Diffusion works faster and higher temperatures, smaller particle sizes, and higher temperature, smaller particle sizes, and greater concentrations. Diffusion is passive. Facilitated diffusion has to have a special channel to get through, but it's still passive. Active transport goes against the concentration gradient. Osmosis is a variation on diffusion. It's the movement of water across the membrane. Water will always go from a high water concentration to a low water concentration. So, and that's the last part, since we went so long last week, we covered more than I intended to. So this is the other half of it. So structures of the cell, or rather the mitosis and cell division, and membrane transport and permeability, along with parts of the microscope and the 4 types of tissues and the skin. So, and that's everything I've got for today. Any questions? Okay, then I'm going to get us out of here, and the test will open up next Tuesday for first lab test. In lab that day, we're going to go ahead and start looking at bones, looking at the skeleton. So, okay, and I will see you all later.